So, very good morning to one and all present here. Myself, Dr. Amit Sharma, welcome you all. Today we have with us Dr. Rupesh Kumar Gautam. He is working as Professor and Head Department of Pharmacology in Indore Institute of Pharmacy. Dr. Rupesh is having 14 years of experience in teaching and research, and he has published 67 research and review paper in Journal of Repute. Dr. Rupesh is having two patent and 42 copyrights, and he has written 10 books, 18 books chapter in different good platforms like Elsewhere, Springer, Bentham, Taylor, and Francis. Dr. Rupesh have guided near about 21 MPharm students for their research work, and he have also organized multiple workshop, conference, FDP at their level. Uh, Dr. Rupesh was the guest editor of the multiple journals as a guest editor. Some of them are phytomedicine, editor of phytomedicine, and second is current pharmaceutical design, neuroscience and behavioral review, evidence-based complementary and alternative medicine, and biomed research international journal. So I welcome you, sir, for the talk.
Good morning, sir. Thank you so much for the invitation. Can I share my slides, sir?
Yes, sir, please, please.
Is it visible, sir? Hello?
Yes, sir,
Hello?
visible, visible. Please carry on.
Okay, sir. Thank you so much, Dr. Amit Sharma, sir, for inviting me. Today's talk is computational approaches in drug discovery. What is the objective of today's talk? To discuss about basics or general idea of computer-aided drug discovery, to discuss about computer-aided drug discovery and in silico pharmacology, to discuss about success stories of CADE, to discuss some useful softwares in pharmacology, and what will be the learning outcome after this talk. Audience able to know about CADE in drug discovery and alternative methods for biological screening techniques. This is the goal, objective and learning outcomes. And now, what is the content of the talk? Introduction, traditional and modern drug discovery, drug design types, CADE in silico pharmacology, CADE advantages, CADE success stories, CADE some tools and databases, some important softwares or alternative biological screening techniques. Introduction. What is the introduction of CADE? On October 5th, in 1981, Fortune magazine published a cover article entitled "The Next Industrial Revolution: Designing Drug by Computed Tomorrow." And after this, Sir James Blake in 1988, Nobel laureate in physiology and medicine, famously stated that the most fruitful basis for the discovery of a new drug is to start with an old drug. And drug discovery development is very expensive and time consuming, 10 to 15 years, and US 15 to 100 million dollar are required for introducing a drug into market. And that's why computer aided approaches are using widely to improve the drug discovery and development pipeline in pharmaceutical research industry. Due to the traditional drug discovery development, expensive time consuming process, CADE was introduced. And this is a traditional life cycle for drug discovery, which takes 10 to 14 years and US dollar 15 to 100 million. This is the first cycle, natural or synthetic compound in pharmacognosy or in chemistry, in natural products. In pharmacognosy हमने study किए होंगे natural products या synthetic compound we can synthesize in chemistry part. And then natural or synthetic compound we will go for the preclinical phase, animal study, rat, mice, rabbit, various model is selected for the various study for diabetes or any other study. We can select the preclinical phase, animal or other animal or other phases are required for the this. And now clinical phase, this is the clinical phase. Clinical trial will be there is the four phases of the clinical trial, that is the, and the last phase is the drug. And now this is the modern life cycle, modern drug discovery design pipeline. The disease cause and mechanism, then target identification, screening, then gate computer-aided drug discovery, preclinical, clinical trials, novel, then in screening method, active compound selection. मैं मिलता हूँ आपसे सर लेक्चर in modern drug discovery, design pipeline, disease that is cause and mechanism, target identification, screening that active compound selection and code computer-aided drug design. This is the two steps are there: SBDD and LBDD, structure-based drug design and ligand-based drug design. And next is the preclinical clinical trial and new drug application. Drug candidate will be introduced in the market. This is a traditional life cycle, and this is the modern drug discovery design. In this, we are using the computer applications. Now, what is the drug design? Intensive process of finding new medications based upon the knowledge of a biological target. Design molecule should be complementary in the shape to the target. वो शेप में होना चाहिए टारगेट के एंड और वो हो सकता है ऑर्गेनिक स्मॉल मॉलिक्यूल या फिर opposite charge to the biomolecular target. And this molecule will interact with target bind. जैसे हम लोग रिसेप्टर में ड्रग जाके रिसेप्टर पे बाइंड होती है, this molecule will interact with target and bind with target, activates or inhibit the functions of a biomolecule such as protein. Now, mechanism based is understand of a disease at molecular level, and drugs can be designed or particularly to interact with the target molecule and disturb the disease. This is the mechanism based drug design, and CADE, which we are understanding today, CADE or in silico or computational methods. Now, what is the CADE? This is the introduction, computer-aided drug design. CADE represents use of computational tools and related resources for drug design, discovery, and development process. Identification of a drug target by using bioinformatics tools. There are very bioinformatics tools are there which are using for identification of drug target and used to identify active drug candidates, other name is hits, and select most likely candidate for further evaluation like leads and optimize leads and transform biologically active compounds into suitable drugs by improving their pharmaceutical, physicochemical, ADMET or PK, ADMET, absorption, distribution, metabolism, excretion, and toxicity, pharmacokinetic properties we are using for this. And now this is the classification of CADE or what is process involved in CADE. CADE two sub-category are there, LBDD and SBDD. LBDD then go for pharmacophore or QSAR, then virtual screening SBDD, then in this binding site identification and docking, then after virtual screening, compound selection, lead optimization, and new drug candidate. Now, types of drug design. These are two types: LBDD and SBDD. LBDD is ligand-based drug design. It relies on knowledge of other molecules that bind to the biological target of interest and used to derive a pharmacophore or molecule which have all necessary structures features to bind to a target active site. Now QSAR, that is quantitative structural activity relationship, is also used, which describes mathematical relationship between calculated properties of the molecule and their experimental determined biological activity. These are ligand-based drug design by using QSAR. We have classified here the pharmacophore QSR, then visual screening and compound selection, lead optimization, new drug candidate. It is the modern cycle. And now, second is the SBDT, that structural based drug design relies on knowledge of 3D structures of the biological target obtained through X-ray crystallography or NMR spectroscopy. Using structures of biological targets, drugs that are predicted to bind to the target designed using interactive graphics, medicinal chemist intelligence, several computational procedures. This is the second type of this structure based drug design. 3D structures of biological are obtained through X-ray crystallography or NMR spectroscopy. Now, structure-based drug design. In this, binding site identification is there. This is the first step in SBDD, structure-based drug design. It relies on identification of surface on protein for binding of a drug molecule that is known as hot spot, जिस पे वो ड्रग बाइंडिंग होगी, ड्रग लाइक रिसेप्टर जैसे बाइंड करता है, that is. This is the structure we can see here. Now, structural based drug design में second part is the docking. That docking क्या है? That is the attempts best matching between two molecule, like right key for the lock. एक lock के लिए जो key होती है, particular lock के लिए एक particular key है, तो that is the matching between the two molecule, that is right key for the lock, the target plus ligand equal to docking. We can see in the figure below that this is the docking in this figure. This is the docking. This is the we have already we have these are the types, that docking, then virtual screening, compound selection, then lead optimization. And now, व्हाट इज द इन सिलिको फार्माकोलॉजी? ट्रेडिशनल मेथड्स में हम लोग क्या करते हैं? स्क्रीनिंग ऑफ नेचुरल सिंथेटिक कंपाउंड बाय इन विवो इन विट्रो मेथड्स, या एनिमल को यूज करेंगे, या लैब के अंदर इन विट्रो मेथड्स से हम लोग कोई भी एंटी कैंसर, एंटी डायबिटिक कोई भी एक्टिविटी उसके निकालते हैं ट्रेडिशनल मेथड्स में, एनिमल्स का यूज करते हुए वेरियस मॉडल को हम फॉलो करते हैं और इसका एक्टिविटी हम ट्रेडिशनल मेथड में निकालते हैं. देन इन सिलिको मेथड्स के अंदर क्या इंक्लूड होगा? दैट डाटा बेसिस, QACR जो हम लोग पहले देखे, फार्माकोफोर्स, होमोलॉजी मॉडल्स एंड अदर मोलिकुलर मॉडलिंग अप्रोचेज, मशीन लर्निंग जो आजकल बहुत ज़्यादा चल रहा है, मशीन लर्निंग, डाटा एनालिसिस टूल्स. तो दीज आर द इन सिलिको मेथड्स इंक्लूड दीज database, QACR, फार्माकोफोर, होमोलॉजी मॉडल्स एंड मशीन लर्निंग एटसेट्रा. Now, what is the advantage of advantage of CAT? That is cost, time कम कंज्यूम करेगा, accuracy is good than traditional method, information of the disease, it provides the best result, and reduction in screening, and less manpower is required, but because it uses the computer. Now, there are various success story involved in the CAT. काफी इस पर रिसर्च हो चुका है, कई सक्सेस स्टोरी भी है. Some examples I am collecting here. That is thrombin inhibitor by using docking or de novo design. Example is heroin. Blood, this is the blood anticoagulant property. Docking और de novo को use करते हुए इनको इसमें अप्लाई किया गया है. Then potassium channel blockers, स्ट्रक्चरल बेस्ड डिस्कवरी लाइक सोटेलॉल, कार्डियोवैस्कुलर सिस्टम ड्रग में आप लोगों ने पढ़ा होगा, आर्टिकुलर एंड वेंट्रिकुलर टेकियारिथमिया. नाउ सम अप्रूव्ड ड्रग यूजिंग CAT, एंजियोटेंसिन कन्वर्टिंग एंजाइम इनहिबिटर, कैप्टोप्रिल फॉर द ट्रीटमेंट ऑफ हाइपरटेंशन. एंजियोटेंसिन कन्वर्टिंग एंजाइम इनहिबिटर जो कैप्टोप्रिल है, दैट इज यूजिंग फॉर द ट्रीटमेंट ऑफ हाइपरटेंशन. Carbonic anhydrase inhibitor, dorzolamide for the treatment of gastritis, macular edema. Renin inhibitor, L-screen, which is used for essential hypertension. And human immunodeficiency virus, HIV protease inhibitor, secunavir, ritonavir, and indinavir for the treatment of HIV. So these are the some approved drug by using CAT. These are the success story behind the CAT. Now there are various drugs are available. I have chosen some example here, and CAT some tools and databases like tools and types, and what is the use of PubMed. All you know the PubMed, all PG or PhD scholars know about the PubMed for the literature search. It is the search engine for searching matter or literature related to medical and sciences for their study. And PubChem, that is a database to collect information about structure and physicochemical properties of chemical compounds. And ChemDraw, chemistry or other students knows about the ChemDraw for the structure part of Chem office to draw chemical molecule, chemical structures को बनाने के लिए इसका यूज करते हैं ChemDraw. And Autodoc, that is software for molecular docking. There are many tools are available. I have selected two. Some here. Now, some important software that is using some alternative biologic screening technique like pass prediction or assist toxicity, Swiss ADME. This is the pass prediction software that pass online predicts over 4,000 kinds of biological activity include pharmacological effects, mechanism of action जो हम लोग pharmacology के अंदर पढ़ते हैं कैडी त्रिपाठी में अलग अलग या कोई और बुक्स के अंदर mechanism of action, toxic and adverse effect, interaction with metabolic enzymes and transporters, influence on gene expression, etc. And to obtain the predicted biological activity profile for compounds, only structure formula is necessary. Thus, prediction is possible even for virtual structure design in computer board not synthesized yet. तो ये हमें प्रॉपर प्रेडिक्शन प्रोवाइड करवाता है ये सॉफ्टवेयर कोई भी केमिकल कंपाउंड है एस्प्रिन और वो भी है तो पहले से प्रेडिक्शन पता चल जाता है उसमें एनिमल्स वगैरह इसकी जो ट्रेडिशनल मेथड में ज़्यादा यूज में आते हैं ज़्यादा उसकी डेथ हो रही तो उस पर ये थोड़ा कंट्रोल करने में तो ये दिस इज द पास ऑनलाइन सॉफ्टवेयर यू कैन सेलेक्ट हियर ये नाउ इस तरह से इसको यूज करेंगे दिस इज द डेमो पास प्रेडिक्शन के अंदर स्टेप्स इन्वॉल्व होगी लाइक फार्मा एक्सपर्ट डॉट आर यू पास ऑनलाइन इसको रजिस्ट्रेशन फ्री है देन स्ट्रक्चर ऑफ अ कंपाउंड फ्रॉम पबकेम और केमरो से आप स्ट्रक्चर कोई भी कंपाउंड का आपको जान पता करना है तो इससे आप लोग स्ट्रक्चर निकाल सकते हैं पबकेम और केमरो से देन इंपोर्ट ऑफ स्ट्रक्चर प्रेडिक्शन ऑफ possible biological activity we can use here also this is the uh, pharma expert online this is the I have already registered here you can registration is free then for the this is the pubchem any structure you can select or smiles or this is the smiles of here or any structure here three predict new compound you can go for the predict new compound here is the three option is there smile small file or marvin js file a you can select then I have selected here the smiles and insert the smile I have pubchem is there I have कॉपी फ्रॉम हियर स्माइल्स किसी भी इसका आप ले सकते हैं एस्प्रिन कोई भी जो कंपाउंड आपको डिटरमाइन करना है देन इंसर्ट द स्पाइस हियर एंड गो फॉर द प्रेडिक्शन कि इसका प्रेडिक्शन कैसे आएगा जो हम लोगों ने एस्प्रिन जो NSAID नॉन स्टेरॉइड एंटी इन्फ्लेमेटरी ड्रग जो इसको देखा है फार्माकोलॉजी के अंदर तो इसका प्रेडिक्शन आएगा या कोई भी कंपाउंड है उसका प्रेडिक्शन चेक कर सकते हैं कि कौन सी एक्टिविटी क्या इसमें ज़्यादा रहेगा तो मैंने क्लिक किया गेट प्रेडिक्शन
हेलो सिद्धार्थ सर
हाँ जी सर
आई थिंक सर डिस्कनेक्ट हो गए ना शायद
कोई नहीं आप मीटिंग को कंटिन्यू रखो सर को कॉल कर लो भी वो कनेक्ट हो जाए
हाँ ठीक है
हेलो
सर यू आर ऑडिबल यू कैन शेयर योर स्क्रीन
ओके सर हम कहाँ तक थे यहाँ तक थे
पास के ऊपर
okay now pass prediction this is the steps involved in pass prediction pharma expert registration is free we can select pubchem or chemdro and import the structure prediction of possible biological activity This is the smiles we have select from PubChem like aspirin or then we can go for the I have selected here, then go for the prediction. This is the possible activity about the aspirin. We have studied in pharmacology that कौन कौन सी एक्टिविटी दे सकता है. तो पहले से हम लोग कोई भी स्ट्रक्चर आप सेलेक्ट कर सकते हैं पबकेम से, एस्पिरिन वगैरह कुछ भी. तो इससे हम लोग ये कर सकते हैं. इसमें 3 ऑप्शन इज अवेलेबल, मोल फाइट देन मार्विन जेएस. आप लोग कोई भी स्ट्रक्चर यहाँ पर कर सकते हैं और देन आप लोग प्रेडिक्ट कर सकते हैं कौन सा ये. तो वो यहाँ पर उसका प्रेडिक्शन आ जाएगा 3 कंपोनेंट या फिर जो भी कंपोनेंट यहाँ पर इज अवेलेबल. तो यहाँ पर आप प्रेडिक्ट कर सकते हैं उसको. अलग अलग कंपाउंड यहाँ पर सेलेक्ट करके यू कैन सेलेक्ट द मोल और स्माइल. ये सब अवेलेबल है. केम ड्रो से भी आप स्ट्रक्चर ड्रॉ करके ये बना सकते हैं. नाउ सेकंड इज द this is the steps involved which we have discussed online also, pass online. Then second step is the registration is free. Then you can select the structure, smiles on any structure, then go for the pubchem se you can select the structure and go for the prediction. Now second is the Oasis toxicity software, that Oasis property explorer lets draw chemical structure and calculate on the fly various drug relevant properties whenever a structure is valid, and prediction results are valued and color coded. जो prediction result आएंगे वो valued होंगे, color coded होंगे. then properties with are shown in red color, showed in red color, where green color indicates drug confirm behavior. तो दिस इज द ओरेसिस टॉक्सिसिटी का इससे पता चलता है हमें. दिस इज द यू कैन सेलेक्ट दिस सॉफ्टवेयर. This is the Oasis software. Though you can download from here, and you can download from here, and it is a free, and then can run. You can run. This is a demo, and this is the Oasis toxicity steps involved. This is the organic chemistry propag and download, setup, and run in computer. And structure of a compound from PubChem or ChemDraw, or you can draw for PubChem, or you can take from the aspirin or any compound you can select from the this and import of a structure and get possible toxicity. उसकी possible toxicity कितनी है वो you can predict from this Oasis toxicity software. जो हम लोग फार्माकोलॉजी के अंदर टॉक्सिसिटी निकालते हैं, एक्यूट और क्रॉनिक टॉक्सिसिटी, उसमें ये Oasis टॉक्सिसिटी सॉफ्टवेयर हेल्प करता है. दिस इज द जब हम लोगों ने ये निकाला और यहाँ पर जब स्ट्रक्चर इसको इंसर्ट किया तो ये आप लोग देख सकते हैं रेड और इस तरह के ये स्ट्रक्चर आते हैं. इससे पता चलता है ये ग्रीन और ये सब बीटा जेनिसिटी और इसका कितना उसका वो है वो रेड उसमें ये साइन हो रहा. तो दिस इज द इम्पोर्टेंट सॉफ्टवेयर फॉर द predict the toxicity. This is the Oasis toxicity software. Then third one is the Swiss ADME. Swiss ADME. This is the also Swiss ADME. This is also software available for free. This is the software you can select for Marvin JS. You can select your or here select any. You can. This is the image or you can go for the and run Swiss ADME. Here is the property you can see here for the molecule. This molecule or part or any compound you can select, and you can this is the property came from the this is Swiss ADME software, and these are the steps involved in that Swiss ADME. Then structure of a compound from PubChem or ChemBro you can select. Here we can select from Marvin Jace is there, and you can insert also. You can insert or you can input here the some smiles also. You can select the smiles from here, PubChem or any, and go run. Then this is the you can see the all property formula and all this. This is the and predict ADME absorption, distribution, metabolic excretion, pharmacokinetic. This is used for compute physicochemical descriptors as well as to predict ADME parameters, pharmacokinetic properties, drug-like nature, and medicinal chemistry friendliness of one or multiple small molecule to support drug discovery. So these are very helpful. So many tools are there. This is we can see here also. This is the first step. And second step, we select the structure. Then this is the outcome. The what is the property of the drug. The various softwares are available. I have explained two or three here only. Now these are the various references we have collected for this study, for this talk, and I have published some one book that computer approaches in drug discovery development and system pharmacology with academic press elsewhere, and one book chapter also with Relay artificial intelligence and machine learning based new drug discovery process and molecular modeling. These are the publication outcome from this case. Now this is some go up and never stop. Then what we are, leap of the faith, make the jump what we want to, क्या बनना चाहते हैं और उसके लिए हमें जंप करना पड़ेगा. बैठे बैठे कुछ नहीं होगा. Go up and never stop. And what is the goal that Swami Vivekananda I follow? Take up one idea, make that one idea for your life. Think of it, dream of it, live on that idea. Take the brain muscles nerves for that idea. Every part of your body be full of that idea, and just leave every other idea alone. This is the way to success, and this is the way great spiritual giants are produced. Other are not talking machines. Now the Abdul Kalam Azad also सपने देखना चाहिए, बड़े बड़े सपने देखना चाहिए. Now thank you so much for patience listening. Now, thank you so much, Professor Dr. D.D. Gupta, Director cum Principal, and Dr. Amish Sharma, Professor and Head, Department of Pharmacy, Practice, ISF College of Pharmacy, Moga, Punjab, for kind invitation, and Sri Arorej Bhatnagar, IRS, Group Advisors, Swiss and experts, and Dr. Gurbi Chawda, Principal, IIB, for the valuable support. Thank you so much. Thank you so much.
Thank you, sir. Thank you for the valuable talk. I hope that all the audience and the student of ISF College of Pharmacy is going to benefit with the information which you have provided. Thank you once again. Thank you. Thanks a lot.
Thank you so much, sir. Thank you.
Okay, sir. Take
Thank
care.
you. Thank you.
Amit sir, you can stop the recording.
Done. कोई और ज्वाइन करने वाले हैं, देख लीजिए कौन है.
रिकॉर्डिंग अमित सर स्टॉप कर देते.
कर दी सर.
ऑन है अभी.
स्टॉप की थी. एक मिनट, जस्ट अ मिनट.